Module 2. Now let's talk a little bit about erythropoiesis, which is the production of red blood cells specifically. The entire process takes approximately 3 to 5 days, and it has several nutritional requirements. For example, you need the right amino acids so that you can build the globin. You need iron ions so that you can complete your hemes and actually bind molecules of oxygen gas. You also require several types of vitamins such as B6, B12, and folic acid. I do want to mention B12 specifically because we will be tying it in when we do the digestive system later in the semester. B12 is typically gained through diet whenever you eat dairy products or meat. However, the B12 that you ingest cannot be absorbed as is through the small intestine. Your stomach produces a chemical called intrinsic factor. B12 must bind first to intrinsic factor within the stomach in order to then be absorbed into your blood through the small intestine. Now, if you have a dietary deficiency of B12 or the inability to produce intrinsic factor, your body is going to be in a vitamin B12 deficiency. A vitamin B12 deficiency actually stops normal cell division within your red bone marrow, specifically the cell division that is occurring during red blood cell production. So not having B12 actually stops your ability to make red blood cells, leading to a dramatic decrease in red blood cell number. This particular type of anemia is called pernicious anemia. Now, during erythropoiesis, there's going to be 4 major developments. First, we are going to have to reduce the size of our cell. Once the cell has reduced in size, we're going to have to increase the number of cells. Once we have all the cells we need, we have to synthesize our hemoglobin. And then once the hemoglobin is made, we have to eject and lose the nucleus and the organelles to eventually have our final red blood cell. So let's talk about the stages of maturation that occur during erythropoiesis. As I mentioned before, it's always going to start with a hemopoietic stem cell. Now imagine you have this stem cell. It undergoes cell division and forms 2 children. One child remains a hemopoietic stem cell. The other child matures and becomes an erythrocyte colony forming unit, or ECFU. ECFUs have receptors for erythropoietin, that hormone from the kidney that has already been mentioned. When erythropoietin binds to the ECFU, it stimulates the ECFU to transform and become an erythroblast. The erythroblasts multiply, so they're going to undergo cell division to increase our cell number, and they're going to synthesize hemoglobin. When the cell division is done, and when all the hemoglobin that is needed is made, these cells are going to eject their nucleus and organelles and become reticulocytes. The reticulocyte is the cell that's actually going to leave the red bone marrow and enter your blood. In about 1 to 2 days, it loses the last of its ribosome clusters and becomes the final mature erythrocyte. So now let's talk a little bit about the iron ions, because they're so important for the transportation of oxygen. You have to make sure that you consume iron daily to replace what you naturally lose. Males on average lose approximately 0.9 milligrams per day. Females lose approximately 1.7 milligrams per day. The main areas of loss are through your urine, your feces, and through bleeding. It is recommended that you consume between 5 to 20 milligrams per day to ensure that you can replace what is lost each day. In pregnant women, it's recommended because there is a fetus gestating who is also forming red blood cells and is in great need of iron. It is recommended in pregnant women that they ingest 20 to 48 milligrams per day to ensure that she can support not just her own red blood cell production, but the fetus's. Now, when you ingest iron through your food, it's a mixture of two different types of iron ions. You have ferric ions, which are the chemical symbol is Fe3+, and ferrous ions, the Fe2+. So you're going to eat a mixture of these two different ions. When the iron ions land in the stomach, stomach acid is going to convert your ferric ions into ferrous ions. Only ferrous ions end up being absorbed. Now, the ferrous ions have to bind to a transport protein made by the stomach called gastroferritin. So gastroferritin is actually produced by the stomach. It's in there with the stomach acid inside the lumen of your stomach, and the ferrous ions are going to bind to it. The gastroferritin can then transport the ferrous ions to the small intestine, where the gastroferritin will release the ferrous ions so that they can be absorbed into your blood. Now, once the ferrous ions are in the blood, it's going to need help with transportation there as well. So once the ferrous ions enter the blood, they're going to bind to a plasma protein called transferrin. Now, once the ferrous ions are bound to transferrin, transferrin's going to transport that iron throughout the body where it's going to be used and or stored. For example, some of the iron ions will be taken back to your red bone marrow, where it's going to be used to make hemoglobin in new red blood cells. Some of the iron ions will be taken to your muscles, where you're going to use it to help make myoglobin. You're going to take some to all of the different cells in the body, because all of your body cells require some iron to make the components of what is called the electron transport chain inside your mitochondria. And the electron transport chain is part of your aerobic respiration inside the mitochondria. So in order to do aerobic respiration, all of your cells need to have some iron. You're also going to take some of the iron ions back to your liver. Your liver actually can store excess ferrous ions. So it's that rainy day supply just in case you need it. What will happen in your liver is the ferrous ions will bind to a protein made by and kept in the liver called apoferritin protein. When the ferrous ions bind to this protein, it forms a complex structure called ferritin, which is your primary iron storage complex. Now, when you start talking about erythrocyte homeostasis, you're talking about your total red blood cell count. And your total red blood cell count is maintained by negative feedback. Now, the controlled condition that's going to be monitored is the concentration of oxygen within your blood. And remember I mentioned hypoxemia? which is an oxygen deficiency inside the blood, well, your kidneys are very sensitive to blood oxygen levels. So when the kidneys detect hypoxemia, they start to produce erythropoietin. The erythropoietin then goes to your red bone marrow. Your red bone marrow then is stimulated to undergo erythropoiesis, which is going to increase your red blood cell count, which is going to increase your rate of oxygen transport. Now, once your blood oxygen levels get back to homeostasis, your kidneys will recognize this and stop producing erythropoietin because you are at homeostasis. Now, I mentioned some causes for hypoxemia earlier, but I wanted to flesh it out a little bit more. Hypoxemia could be caused by blood loss, so just losing blood volume because you've been injured. However, you could have hypoxemia because the air that you're breathing has a very low oxygen partial pressure, so there's not as much oxygen in the air you're breathing. For example, if you're living at high altitudes. However, the air you breathe could be fine. Another problem you could be having if you are hypoxemic is that you have poor gas exchange within the lungs. So you're breathing in enough oxygen, but you don't have sufficient oxygen diffusion within the lungs, and your blood is oxygen deficient. Now, the air you breathe could be fine, your lungs could be fine, but you could have anemia. So either not enough hemoglobin within your red blood cells, or just not enough with regards to red blood cells. However, you could have a patient where the air they're breathing is fine, their lungs are fine, they're not anemic. And what you might be looking at is just poor blood flow to the kidney itself. Your kidneys have no way of knowing what is causing the oxygen deficiency. They just recognize oxygen deficiency. So if you have a partial blockage heading in the renal artery, which is heading to your kidney, that decrease in blood flow is going to appear to the kidney as you have oxygen deficiency throughout the whole body. And your kidney will respond in the same fashion as it would with any of the other possible changes. Now, I mentioned erythrocytes have a lifespan. They live 120 days, and you clearly have a lot of them. So imagine that you are having all of these red blood cells dying, and you are forming new ones at the exact same rate that they are dying, so you maintain those nice homeostatic levels. But if you're breaking down billions of red blood cells per day, where does it happen? You're not just accumulating dead cells within your blood. So we need to talk about when the erythrocytes die, where does it happen? What is done with them? And the primary disposal areas for old and damaged erythrocytes are your liver and spleen. These two particular organs are packed with macrophages. Macrophages are phagocytes. They have the ability to eat and destroy pathogens, debris, and old cells. So as your blood is flowing through both of these organs, the old red blood cells, the damaged red blood cells, are actually pulled out of your blood and are completely broken down. Now, it's not a perfect process. For example, you could have a patient at some point in your career who is suffering from hemoglobinuria. Hemoglobinuria is when you actually have hemoglobin showing up in your urine. And in a healthy individual, this does not happen. In a healthy individual, the hemoglobin is going to be broken down in the liver and in the spleen, and you won't have free hemoglobin floating around in your blood to be filtered out at the kidney into the urine. However, if your phagocytosis does not occur appropriately, your red blood cells that are old can be rupturing, and then that hemoglobin is just kind of overflowing into the blood and into your urine. The urine itself would appear red because hemoglobin's a protein, it's not alive. As long as it's exposed to oxygen, those little hemes will grab onto oxygen gas molecules. So as your urine is leaving the body, it's passing through the air in the toilet bowl. And all those little hemes are going to be grabbing onto all of that oxygen, turning the hemoglobin protein in the urine to that nice little cherry red. So it actually is going to look like there's blood in the urine. Now, hematuria is slightly different. Hematuria is when you have red blood cells present in urine. Now, normally, because of the filtration structure inside the kidneys, formed elements are normally not found in urine. So having red blood cells actually in urine is indicating that something is wrong. However, it's not a problem with the liver or the spleen, as would be the case with hemoglobinuria. In hematuria, you're actually looking at some type of damage to the kidney itself that has caused bleeding, allowing red blood cells to pass into the urine, or perhaps some blood vessel damage in the urinary tract itself. Now, when you have a 120-day-old red blood cell and it's passing through the liver or the spleen, and it's pulled out and a macrophage goes to act on it, the actual process of disposal is called hemolysis. In hemolysis, the red blood cells are going to first rupture, so the plasma membrane is going to pull apart, and the contents of the red blood cell are going to spill out, and the hemoglobin is going to be released. Once hemolysis, that rupturing and content spilling out, is accomplished, then your liver and spleen are going to have to do some recycling because you have all of that hemoglobin that's now been released inside these structures. Now, as the hemoglobin is broken down, you have to remember that it's got two main components. You have globin and the heme. And even with that heme, you have two components. You have the iron ion and you have the non-protein pigment. So the globin, the iron ion, and the non-protein pigment are all going to be handled differently. Now, the globin itself is going to be broken down into free amino acids. And these amino acids will pass into your blood and actually be used anywhere in the human body that they need to be used. So it's quite logical to assume that some of the amino acids, in fact, probably a good number of the amino acids building your hair and your fingernails, used to be part of hemoglobin inside a red blood cell somewhere. So the globin completely and totally recycled and reused. Then you deal with the heme. Well, the iron ion is going to be retained and recycled. The iron ion is going to be released into your blood, where it will then bind to transferrin and get reused as described previously somewhere in the body. The non-protein pigment, however, is different. The non-protein pigment is not recycled and reused. It is a discarded waste product. So your macrophages are going to act on that non-protein pigment. And they're going to change that nice reddish non-protein pigment first into another pigment called biliverdin. Biliverdin has a greenish color to it. So we're going to go from the nice red heme to the greenish biliverdin. The macrophages will continue to act on the pigment and change the biliverdin into another pigment called bilirubin. Bilirubin has a yellowish color. Now, this bilirubin, if it is being produced in the spleen, it's going to bind to albumins in the blood and get transported to the liver. Your liver will pull the bilirubin out of the blood, combine it with the bilirubin that the liver itself has accumulated because it's also doing the recycling, and all of the bilirubin is going to be placed into the main secretion of the liver called bile. Now, bile will be discussed in the digestive system because it contains a component that's going to be very helpful for digesting and absorbing your lipids. However, for right now, just know that by putting the by putting the bilirubin into your bile, your liver now can actually dispose of it. It can get rid of this waste product because your liver is going to use a series of ducts to transport this bile into your small intestine. Now, once the bile is in the small intestine, I'm sure you've heard that you've got bacteria that reside inside your alimentary canal, inside your intestines. And these bacteria are going to start to break down the bilirubin. And they're going to break it down into two components, urobilinogen and stercobilinogen. Now, these two components are going to continue to be broken down even more, and the urobilinogen is going to be broken down into urobilin, and the stercobilinogen is going to be broken down into stercobilin. Now, urobilin, some of it is reabsorbed from the small intestine and back into your blood. Now, urobilin does have a yellow color, and when urobilin passes through the kidney with your blood flow, the urobilin is actually filtered out of your blood in the kidney and it's placed into your urine. So when you urinate and you see that nice yellow color to your urine, you're actually looking at the urobilin from your red blood cell breakdown. So the next time you use the bathroom, as long as you see that nice yellow color, you know that your body has successfully broken down red blood cells that day. Now, some of the urobilin does stay in the small intestine, and all of the stercobilin remains in the small intestine. Now, the funny thing about stercobilin is that it has a different color. It's actually brown. So, as your food is passing through the small intestine and then through the large intestine, the urobilin and stercobilin remain in the intestines. And when you defecate, when you have a bowel movement and your feces emerge, they have that really nice brown, sometimes yellow-brown color. And again, that color is coming from the successful breakdown of your red blood cells in both your liver and your spleen. All right, now that you have an orientation to your red blood cells, let's take a look at our white blood cells. First of all, how are whites and reds different? Well, your red blood cells had that biconcave shape. Your white blood cells, on the other hand, are spherical. So imagine a golf ball, a ping pong ball, a baseball, just a lot smaller. Your red blood cells live an average of 120 days. White blood cells have varying lifespans. Some white blood cells might only live for a few days. Others can last for years, even potentially an entire lifetime. Red blood cells did not have nuclei or organelles. White blood cells have both. They have nuclei and they have the full accompaniment of organelles, meaning they can carry on extensive metabolism and they can repair. Your red blood cells had 33% of their mass composed of hemoglobin. Your white blood cells have no hemoglobin, completely missing it. Your red blood cells were the most numerous formed element. Your white blood cells are actually less than 1% of your blood volume. They're outnumbered by your red blood cells easily 1,000 to 1. So your white blood cells in a healthy person range between only 5,000 to 10,000 per microliter. Now, you have 5 different types of white blood cells, and they are broken down into 2 classes based on whether or not they have visible granules in their cytoplasm. These granules, if they have them, will appear as darkly stained clusters. Sometimes people say they look like big grains of sand, grains of rice. Their cytoplasm will look chunky. I've even had people say that the cytoplasm looks chunky. So if you're classifying your white blood cells, the first class are called granulocytes. Granulocytes do have visible specific granules in their cytoplasm, and there are 3 different granulocytes. You have neutrophils, eosinophils, and basophils. The A granulocytes, whenever you have the letter A placed at the beginning of a term, it's meaning something is absent or it's missing. So A granulocytes have no visible specific granules in their cytoplasm. And the A granulocytes are your monocytes and your lymphocytes. Now, in the first lab that you're doing this semester, the blood lab, part of the actual lab experience is to be able to visibly identify the 5 different types of white blood cells, in addition to red blood cells and platelets. But also, you're responsible for filling in an informational table about the 5 types of white blood cells, including their overall functions. And you're to fill this table in using the Saladin textbook. So, because their functions are part of your lab activity, I'm not going to re-lecture on their individual functions at this time. But no, since you are studying the functions for lab, those functions can show up on lecture exam 1 as well. Now, when you start thinking about white blood cells, their overall function for the body is defense. Okay? Their job is to make sure that if a pathogen comes in, that it's dealt with without causing disease. If you encounter a toxin, it's dealt with before it causes a disease or a disorder. So, they have to be able to respond to those particular things. The problem is that the white blood cells are being transported inside a blood vessel, but these pathogens and toxins are actually located outside the blood vessel in your body tissues. So the question remains is, how do your white blood cells arrive where they're needed? So in order to accomplish that, your white blood cells are going to have to be able to do a few things. For example, they're going to have to be able to migrate out of your bloodstream. Now, migrating out of the bloodstream is going to be a 2-step process. Because remember, you have constant blood flow through every blood vessel in your body at all times. So the first thing that your white blood cell is going to have to be able to do is it's going to have to find a way to attach itself to the wall of your blood vessel in order to get out into the tissue where the potential infectious material is located. So the first step is margination. Margination is when your white blood vessel adheres or sticks to your vessel wall. Now that's great. Your white blood cell at least is clinging to the vessel wall in an area where there is something it needs to help with. But it's still useless because the inflammation or allergen or pathogen is not in that vessel. So once your white blood vessel has attached to the wall, it's going to have to undergo emigration. Emigration is also referred to as diapedesis. And because I have trouble saying it, we're going to stick with emigration. A lot easier to say. If you can say it, you can spell it. Now, when your white blood cell undergoes emigration, it actually can squeeze itself in between the endothelial cells that are forming the wall of your capillary, your blood vessel. And you can even see in the figure at the bottom a white blood cell squeezing its little body out from between the cells forming that wall. So migrating out of the bloodstream involves margination and emigration, a fancy biological way of saying it has to stick and it has to squeeze. All right, so step one accomplished. However, your little white blood cell now has to be able to move and actually travel to where it is needed, which means that we're going to have some type of mobility. And your little white blood cells are going to have what's called amoeboid movement, meaning they have the ability to cause a gliding movement of their cytoplasm. And when they shift their cytoplasm, they have the ability to shift the position of the entire cell. So imagine them sort of creeping and crawling in between all of the other cells in your tissue, trying to get to where they're needed. Now, cells don't have eyes. The inside of your body, kind of dark, okay, don't have a light source in there. So how do the white blood cells actually know where to go? I mean, it's one thing to be able to get out of your blood and into tissue and be able to move, but how do you know which direction to move in? And that's where your white blood cells actually are going to follow chemical stimuli. They're going to do something called positive chemotaxis. In positive chemotaxis, the white blood cells are actually going to follow a chemical trail back to its source. So it's sort of like moving up a concentration gradient, where you get a little whiff of the chemical and you start moving in its direction, and then the chemical gets a little bit stronger and you keep moving in that direction, and eventually you get to the source of the chemical, where you have that highest concentration, and that is where your white blood cell needs to act. Now, some of the chemicals white blood cells like to follow are different microbial toxins. Also, your damaged tissues do produce chemicals called kinins. So if you had some type of trauma, for example, you had the nice little, I guess, splinter pierce your skin. It's got bacteria on it that might be releasing some toxins. You had tissue trauma, you're releasing kinins. All of these chemicals would help to attract your white blood cells. Now, once your white blood cells get where they need to go, as you have seen in the very first lab from filling in your table, they all do different roles when they get to where they need to be. For example, your neutrophils, your eosinophils, and your monocytes all have the ability to do phagocytosis. So they have the ability to eat and destroy the different pathogens and toxins that are in that area of trauma. So white blood cells have to accomplish a lot of different small goals in order to be able to actually even do their defensive function in the body. Now, as a clinical professional, you're going to be dealing with sick people on occasion. And if a doctor suspects a viral infection, parasitic infection, bacterial infection, allergic reactions that are ongoing, and he has to try to, or she needs to narrow down what could possibly be wrong with the patient, he might order what is called a differential white blood cell count. A differential white blood cell count is a count of each of the 5 types of white blood cells in a sample of 100 white blood cells. So what you would do is you would take a 50 microliters of blood from a patient sample. You would basically smear that blood across a microscope slide, stain it so that you could distinguish the different cell types that were there, and then under a microscope scan through that blood smear looking for 100 different white blood cells. And every time you find one, determine which of the 5 types it is and record it. And what you would end up with is percentages of how many white blood cells in your sample were neutrophils, how many were eosinophils, how many were basophils, how many were monocytes, how many were lymphocytes. Now, in a healthy person, the neutrophil percentage is usually 60 to 70%. So these are your highest percentage white blood cell type. Your second highest white blood cell type are actually your lymphocytes. They range in between 25 to 33% in a healthy person. Your number 3 in abundance are your monocytes. They range usually 3 to 8%. Eosinophils rank in at number 4, only about 2 to 4%. And basophils are the least abundant leukocyte. They're the least abundant white blood cell, coming in at less than 0.5%. Now, these are the percentages in a healthy person. If your patient is ill, these percentages could be different. And since each white blood cell type has its own particular role in defending you, you can use that information to narrow down what's wrong. For example, eosinophils are used to help battle parasitic infections. So if you have a parasitic infection going on, your physician would take a look at your differential white blood cell count. They'd see not 2 to 4%, but maybe they're going to see 15% eosinophils. And what they'll say to themselves is, okay, we've got a higher eosinophil percentage that's possibly indicating a parasitic infection or an allergic reaction happening in the patient, and it just narrows down their search for what's actually wrong by using that little clue. So a differential white blood cell count is a really important diagnostic tool. You can detect inflammation, infection. You can look at the effects of poisoning by different chemicals. You can monitor the effects of chemotherapy on your ability to produce white blood cells. You can monitor specific blood disorders. You can detect allergic reactions, parasitic infections. So it's really important information clinically. Now, if you have that blood smear and you count or you have a blood sample, you can do what is called a total white blood cell count. So you can just look at how many white blood cells are actually in the whole blood of your patient. And remember, in a healthy person, it's usually between 5,000 to 10,000 white blood cells per microliter. However, if you do that total white blood cell count and you have a value over or greater than 10,000 per microliter, you're looking at what is called leukocytosis. So leukocytosis means your count is well over 10,000 per microliter. This leukocytosis is a natural protective response that occurs within the body in response to stress. So if you've got a microbial infection, if you've had surgery, if you have been subjected to anesthesia, even exercise will do it. So anytime the body is put under any kind of stress where you have the potential need to defend yourself, your body will be stimulated to increase its total white blood cell count. What you never want is you don't want leukopenia. Leukopenia is shown when your total white blood cell count is below 5,000 per microliter. And what that means is that you now have impaired body defense. You don't have the right number of white blood cells to be able to protect you if something tries to invade. Now, leukopenia can be caused by metals, toxic metals that you can encounter in your environment, like lead, arsenic, and mercury, radiation treatments, and chemotherapy treatments that you would have fighting cancer. They cause leukopenia, which is why when someone is undergoing chemotherapy, they need to be more cautious when they're out in public, take more precautions to protect themselves from exposure to bacteria and viruses because they're more susceptible. Their defenses are lower. Also, diseases like the measles, the mumps, chickenpox, AIDS, polio, influenza, typhoid, all of these viruses also decrease your ability to produce white blood cells, which one makes them harder to treat, but also then opens up a door for secondary infections. Whenever you have the flu, you need to make sure you're monitoring yourself because pneumonia is a really common secondary infection that can occur after you've had the flu. And that's because the flu reduces your ability to produce white blood cells so that if the pneumonia pathogen enters your lungs, you have impaired ability to defend yourself against it. So leukopenia is not something that you want. All right, now let's talk a little bit about leukopoiesis, the formation of your white blood cells. Now, leukopoiesis is going to begin with that nice hemopoietic stem cell, and the hemopoietic stem cell is going to undergo cell division, and one of its children will remain a stem cell. The other child will become a colony-forming unit for one of the white blood cells based upon the chemicals in the environment. These colony-forming units will continue to develop and form precursor cells like myoblasts, monoblasts, and lymphoblasts. Now, the myeloblasts, as they continue to develop, they can lead to the formation of a neutrophil, an eosinophil, or a basophil. The monoblasts, as they develop, only lead to the formation of monocytes. and lymphoblasts only lead to the development of lymphocytes. Now, as you can see on the image on the screen, there are 3 different possible lymphocytes that can be produced. You have B lymphocytes, T lymphocytes, and natural killer cells. Natural killer cells are usually abbreviated NK cells. We'll be talking more about these 3 different types of lymphocytes and what they do in the lymphoid chapter. Now, at the beginning of Chapter 18, so in Module 1, I mentioned that there were 2 different types of hemopoiesis, myeloid hemopoiesis and lymphoid hemopoiesis. In myeloid hemopoiesis, your white blood cells are born completely mature in your red bone marrow. So all 7 types of white blood cells can be born and fully develop in the red bone marrow. Lymphoid hemopoiesis is different. Lymphoid hemopoiesis does begin in the red bone marrow. However, the cells can't fully develop here. So with lymphoid hemopoiesis, which is leading to the formation of lymphocytes, the cells are born in the red bone marrow, but then they have to leave your red bone marrow, they have to travel through your blood and end up in an organ called your thymus in order to complete development. Now, lymphoid hemopoiesis is most important for the development of your T lymphocytes. But again, this detailed information of how they're formed and what happens is going to be covered in your lymphoid chapter. So for this exam, just know that lymphoid hemopoiesis begins in your red bone marrow, but to complete development, the cells, especially your T lymphocytes, have to leave your red bone marrow and complete development in the thymus.